Hello, welcome to this video. I will be sharing you the beginner's guide to structural bioinformatics. This tutorial will be split into three different sessions, and this will be the first of the three parts sessions. And the materials for this tutorial is available in the description box under this video. Now, structural bioinformatics is a study of predicting the 3D structures of biological macromolecules. These can be proteins, nucleic acids, and the complexes that are formed by them. It can be useful for us to screen large molecule of compounds in a large molecule of compound. If we want to do a drug design or pesticide research, we can modify certain residue by performing in silico mutagenesis. So we can find what are the residue that be important for us to do in vitro mutation and follow-up analysis. It is very useful to predict a novel protein structure and predicting its potential activities. So this process can help to accelerate the pattern acquisition process. This is what the third point is what most people would be interested in. We can generate in silico paper for free, and also we can enhance other in vitro and vivo studies. So molecular docking, we are predicting the preferred orientation of a molecule. So molecular docking, we are predicting the preferred orientation of a molecule that is called ligand to a second molecule that we call receptor when they are bound to each other. So when we're talking about receptor, we tend to be meaning the protein, but it can really depends on what you're looking at. And the ligand is usually the smaller molecule that will bind to the receptor. So ligand can be small compounds, it can be lipids, it can be proteins, and it can even be nucleic acids. And what a molecular docking software will do, it will rank the dock position based on the ligand binding affinity in terms of kilocalorie per mole. Um, to put us in the correct context, when a ligand binding affinity is stronger, the value in kilocalorie per mole would be more negative. These are the molecular docking tools that can be used. It can be protein small molecule. If you are looking into the protein small molecule, then we can use something like Autodoc 4 or Vina, which are the most popular. You can explore other molecular docking softwares. For this tutorial, we'll be focusing on CBDoc2. And if you're working with protein-protein interactions or typically a large molecule, we have other molecular docking tools such as HDoc or HeadDoc, and so on and so forth. Now, molecular dynamic simulation is again a simulation of physical movement of molecule within a system called a solution over a particular time period. Molecular dynamic simulation is a much in-depth analysis of the protein-ligand interactions. When we're talking about molecular docking, we're analyzing the basic interaction between a small molecule or a ligand with the receptor and nothing else. But when we're doing a molecular dynamic simulation, we are performing under usually a solution. So they are factoring in the the hydrogen bonding effects of the water molecules that are present within the system. So these water molecules can interact with our receptor and also ligand molecules, causing them to change shapes. And these changes can affect the stability of the interactions. Molecular docking is essentially a molecular docking is a very quick but basic processes, and molecular dynamic simulation is a more laborious process, but the data output is very important. And when we're doing a research, we typically perform molecular docking first to predict the dock position, and then we validate the dock position through performing molecular dynamic simulation analysis. After that, um, this particular study will actually be looking into this particular research. And a bit of introduction to the IBCC acid receptor. We're looking into the pyrobacterium resistant 1 like protein. So it activates an ABA responsive genes. What activates the ABA responsive genes is the SNRK2. It is colored in green circle, as shown in the figure. Okay, if there is no abscisic acid ABA, then the protein phosphatase 2C, PP2C, will an inhibitor to SNRK2 will continue to bind to SNRK2, inhibiting the SNRK2 activity, so the ABA signaling becomes off. But if we have many presence of abscisic acid, the abscisic acid will bind to the PYL, and this PYL AB complex will will be able to bind to the protein phosphatase 2C competitively. So it will outcompete the SNRK2. So SNRK2 becoming available and to be phosphorylated, and become phosphorylated, it can activate the ABA signaling processes. So the study that we're going to perform is to characterize the POL receptor. Okay, the flow of study is first we're going to acquire a sequence of interest, and then we're going to model the sequence. We're changing the sequence from one length of amino acid string into a folded three-dimensional protein. Once we got the protein, we're going to try to identify or predict the ligand interacting site, and then we'll perform molecular docking, and we'll also perform mutation analysis as a bonus of this study. For part one, we will be focusing on this particular part. Okay, firstly, we're going to acquire the sequence of interest. Now, when we're looking into the sequence of interest, we can have multiple different sources. If we're performing a protein preferential study or an natural product analysis, we can purify a product of interest that we can send for sequence, and then from the sequence, we can try to predict the 3D structure. So it can be from your own samples. If you're interested in a particular study, you can refer to research paper that reported the protein sequences. Usually they will be reporting in terms of the gene sequence, the gene codes from the NCBI. So you can use that as a reference to find the protein that you want to study further. If it's not available from previous research article that you have known, we can actually mine the proteins from the NCBI. For this practice tutorial, we're going to perform mining the receptors from NCBI. So simply, we're going to open NCBI taxonomy browser. We're going to go for the interest records protein. We can enter the name of protein of interest, which is the abscisic receptor, and then we can select as-many-if-one. For the self-tutorial, we're going to download everything. We're going to download them as FASTA. Now let's try it out. So firstly, we're going to open our internet browser. So let's go with NCBI, .gov. Okay, for the sake of this study, let's look for maize. So we're going to write here, we call from the all databases. We go for taxonomy, and then we're going to write Zia maize. From here, we can click like this, and then we can just continue clicking this particular Zia maize. So this is a taxonomy browser for Zia maize, which is corn. If you are interested in the nucleotide sequence, we're going to look for the nucleotide. If in our tutorial we are interested in the protein because we want to characterize the protein structure, so we're going to select the protein here. Okay, when we are performing our search, it is important for us to leave this taxonomy ID here, and let's just search our protein of interest, abscisic acid, abscisic acid receptor. And we click on search. Now we can see we have Xiamis over here. For our tutorial, let's assume we want everything. So we're going to download them as a list of FASTA sequence. To download the list, we're going to click on Send to, click on File, Format, change from Summary to FASTA. You can sort in any order you like. Just leave as default if you do not know which ones you want. And we can click on Create File. So this is the the FASTA sequence that the NACBI give us. We save as. Okay, let's just work with our bioinformatics workshop folder and write them pure maze URL sequence. So let's open list. Okay, let's check it out. So as you can see, there are the sequence of the MCS receptors that are given from the NCBI. So now we have we have now known the sequence. So now we're going to model the sequence. There are several options of protein modeling softwares. This is just my personal preference. If you are working with a monomeric protein, it is better to use either TR Rosetta or Swiss model modeling software. If your protein is dimeric or it is a very novel sequence, so basically like you got it from a very novel species out from the wild, something like from Antarctica, it is better to use AlphaFold 3. And for this exercise, we're going to model our sequence using TR Rosetta. We're going to copy this protein sequence from the FASTA files, and we're going to submit the PYL10 sequence. However, in practice, we should model the sequence using different modeling softwares and evaluate them using different evaluation servers. We're not going to, I'm not going to explain in detail what this does. This is for you to refer on your own. But the idea is they will give a particular scores, and what scores is considered good is actually dependent upon past papers. But alternatively, you if you know a reference protein that is similar to your own protein of interest, we can perform the evaluation for that reference protein as well and compare the scores between the reference protein and also our model protein of interest. If the score is very similar, then we consider it to be good. Right? So let's do this now. SBI. Let's just go to, we can actually type in TR Rosetta. Then it is from the Young Lab web server. From here, it's very simple. We're going to paste our sequence here. So let's grab our sequence. Oh yeah, I forgot. I have that on notepad. So let's just use the top sequence for the sake of it. So we're going to Control C to copy, and then we're going to paste it here. We can Control V or right click and paste. Um, right. When you performing any work that using a web server, it's a good practice to put in your email here. So when you do your work, it is important for you to leave your email here. But for my own, I'm just going to leave it blank because it's just a practice. And also we have the option about not using template. You want to perform blind modeling. This can also work for TR setup. But for us, if you're using maze, it is more likely that you have a very similar homologs from Arabidopsis or rice or so on. So it's better to use templates for our case. So we can now submit. So yep, we can check the model results here when it's done. All right. So when it is done, I'll get back to you again. So this is the results of our TR Rosetta modeling. This is quality of our protein. The predicted LDDT, basically, the more of the position that is higher than 80, the better. We can report the TM score. This is what matters the most. And then we can download the model. Also we give five models, but we just download the first one, model one, and then we can click save as, save into our Bioinfo workshop folder, rename it as PYL10, and we are done. Okay, now since we already got the means PYL10 model, we're going to try to identify or predict the ligand interacting site and subsequently perform the molecular docking. Okay, when we want to find the ligand binding site, there are two broad ways of doing it. Firstly, we can compare with the known structures, or we can do blind docking. So if you read, if from reading, you know a protein of similar structure, so we can manually search those proteins from the protein data bank. So in our case, we can try search for example, Arabidopsis thaliana, or maybe a very similar species such as rice. So you can find rice abscisic acid if it's available from the protein data bank. But if you want to be more methodological about this, we can submit our protein structure to the DALI server. So what DALI server does is that it will find, perform structure similarity against all the proteins from the protein data bank, and then give us proteins that is most similar to our query protein, which is our protein of interest. Alternatively, you can submit your sequence, not the 3D structure, of the PYL10, and we perform PSI-BLAST against the protein data bank. PSI-BLAST is a bit different from the normal BLAST-P because PSI-BLAST is more towards finding a structural similarity. This is a very surface explanation about what it is. The truth is much more complex, but I'm not going to delve into it in this tutorial. It's more of a how to use it rather than explaining about what it does. But ideally, when we want to find out the known structures, we want the reference to have a ligand bound to. So if we have a reference protein that are complex with a particular ligand, so we know where the active site is going to be. So we can simply superimpose our structured proteins of interest onto the reference protein, and then we can have some idea about what's the binding sites going, where the binding is most likely going to be. And alternatively, we can perform what we call blind docking. Usually, a molecular docking web server have their own algorithm to predict any ligand site. For this practice, we're going to perform CB.2. We can go to this website, and then if the protein of the reference protein does not have a ligand bound to it, we can repeat this process by performing CB.2 for the reference protein as well. So let's try it out. Okay, let's first go to the DALI server here. So from here, we're going to click on PDB search, and then we're going to find our file. Here, we're going to upload our PYL10 model protein. Open, and then you can leave the job title here. And but it's good practice to put your email on the email box, then click job submit. Right. So we got it quickly. So we want to match. Ideally, we want to find matches against the PDB90. What this PDB90 means is that um, so we want to. So from the DALI survey results, we have matches against PDB 25, 50, 90, and full PDB. 25 meaning it find the matches that shares no more than 25% sequence similarity. PDB50 is no more than 50% sequence similarity. PDB90 is no more than 90% sequence similarity, and full PDB is they don't have any criteria. um, for practice, let's just find the matches against PDB90 because it shows a very good structural similarity. And from here, we can see that the output is sorted by the Z score, and it appears to give the highest Z score to as the number one. And as you can see, the RMSD is very low for 5UJVA, and 0.3 RMSD is actually a very almost identical structure essentially. So we can click here to find the structure of our protein of our very closely similar proteins. Oh, sorry, my bad. We probably have to go to the PDB website. So let's go to PDB website, but let's just copy 5UJV. But note that if it is written 5UJV-A. So first let's check it out. 5UJV PDB. And let's click on the structure. Okay, so it is a crystal structure of iron PYR1 in complex with abscisic acid. So it is an abscisic acid binding protein, and it is complex with abscisic acid, so it's very good. So our crystal structure has a abscisic acid bound to it, and when our see how our receptor is 5UJV-A. Dash A means it refers to the chain A of the structure. So we're going to download this as our reference protein. So we're going to click download files, download as PDB format, and then we're going to save it in the workshop. For easy reference later on, I'm just to going write 5UJV underscore reference. Right. So we're going to open this file. Okay, this PDB file can be opened using BioVia Discovery Studio. I'm going to show you the website. So you can download it from here. Alright, but you're going to have to register. Once you have registered, and then you got the free license, so you can open the files using the you can open your PDB files using BioVia. Okay, here's the structure of 5UJV. Okay, we can view each one by pressing Ctrl H to open up the hierarchy panel. Alternatively, you can click View and click on Hierarchy here, and it will show this particular panel. If we highlight A, see how it is highlight here. I'm going to press again. Yeah, and then we're going to write here as well. Okay. If you click on if we click on this button here, it's going to show you the amino acids residue. And if you're highlighting by holding shift, you can highlight a particular segment of the protein. It will be highlighted in yellow. If you want to create like different places, you can hold control and click on the sequence that you wanted to label. So yeah, and then you can change color or do whatever you want, but the coloring part is not going to be the focus of this particular tutorial. There are other tutorial that you can look on YouTube on how to present your structure better. But what we can see is that the first one is the proteins. Let's see, you see, and the second one appears to be the abscisic acid. Yep, that's A8S900. I think this will be, yep, that's the abscisic acid for the chain B, and that's abscisic acid for chain C. And A here appears to be like dotted. So let's check it out. Okay, so this one is HOH. HOH is some water molecules. And depending on what molecular docking software you will be using, you will want to remove the water from our structure before we submit them for analysis, because if the molecular docking software does not remove these water molecules, it may possibly interfere with the ligand accessibility, so the docking might not be accurate, right? So for this tutorial, we want to keep only the chain A. The way to do it is by simply we click on new. It will open a new molecule window. So we go back to 5UJV. Let's just select by holding down control. We select only chain A, but we do not want water, so we just click on the top one and the second one. We click on Control C to copy, and then we go to the new molecule window. We click on it like this, so it's highlighted. Then we Control V to paste them. So this is the structure of chain A only, and this will be our reference for our PYL10. So let's save this. I'm going to click on Save As. All right. So from here, we're going to save them as PDB or protein data bank files here. Let's give it name as 5UJV pro, chain A only. I love naming it like this. You can name it however you want. Okay, so what we have done is actually this particular step. We superimpose PYL10 to our reference protein before running the CB.2. We load the minimized AB ligand to the ligand box. We load PYL10 in the protein box. When then we click on auto blind docking, and then we download all the complex results. We open the CB.2 folder. We combine all of that complex using BioVia, and we select the dock ligand that coincide with the reference ligand position. And again, if our reference protein does not have a complex ligand, we repeat the CB.2 for the reference protein. Okay. Now, after minimization, we're going to perform superimpose of our PYL10 to our 5UJV chain A only structure. We're going to use Chimera. Let me check. The installation for the Chimera, you can use Chimera or Chimera X. Chimera without the X is a bit more difficult to use, but things tend to work better in normal camera compared to camera X. Both of them are available under the software folder, but just going to start showing you the camera. I'm just going to use the camera without the X version. Okay, let's open the file. So we're going to open PYL10. Then we go open 5UJV Chain 8 only. Right, so that's how it looks. You can click on tools, and then we go for structure comparison, and we click on matchmaker. Now, either way is fine, really. You can match against, you can have the reference structure, the 5UJV, and the structure to match against PYL10, and then, or vice versa. Yeah, it doesn't really matter. So, but the most important thing is we want them to be superimposed together. Okay. So now we're going to save the PYL10 structure. Oh, sorry, my bad. Um, we're going to save the PYL10 and also the reference protein separately. So this is how we're going to do it. We're going to click on save file, save PDB. And firstly, let's save the PYL10 and give a new name, PYL10 prep. Save relative to, it's better to change it to our reference protein and click on save. And then we're going to do the same for our reference protein 5 UGV. Let's give another name, A this, and then we're going to save this model and yep, keep it similar to the relative to 5 UGV chain A only. Okay, if it's done correctly, we're going to have these two files and we're going to upload the files into CBDoc2. Now we have our proteins ready and also our ligand ready. We can upload to CBDoc2. But in our case, our reference protein already have a ligand bind to it, so we do not have to do our reference protein molecular docking or CBDoc2. We just have to do PYL10. If our reference protein does not have ligand bind to it, we're going to have to do CBDoc2 for our reference protein as well. So let's just go to the CB.2 website. You can just Google or opening from the PowerPoint link. And we're going to drag and drop our protein and also ligand. Okay, let's just drop our POL10 prep into here. And then we're going to drag and drop the AB ligand, minimize here. Again, it's a good practice for you to write your email. But I'm not going to do for this practice. And now the process is running. I'm going to pause and continue once it is completed. Now it is complete. We're going to click on view results. Okay, the output is differentiated into the structure-based blind docking and also template-based blind docking. So the structure-based blind docking, it used the algorithm to infer which one's the ligand binding cavity, and CBDoc will give you the top 5 potential protein ligand binding site. And also, we have template-based blind-based docking, where it looks for protein of similar structure and use their information of ligand interaction to predict where the ligand binding cavity is. What you can see here, we have only 5 pockets instead of 6, because we should have 5 structure base and 1 template base. The reason being is that the first cavity, the first pocket, appears to have the template. So the first, this data or the template is coincide with the C1 pocket. So this is very likely to be the actual protein ligand binding site. But in practice, let's just download each one file, because this will show different ligand binding position on our protein. So we're going to download one by one. Make sure we download into the Bioinformatics workshop. Just keep them long as this. We can make it shorter, something like this. Like to do like this. Make it shorter like this and add C to it, so I know which one I'm looking at. And then copy this. Okay, so we've downloaded all 5 of them. And before we finish, I want to share the download queue pocket. The download queue pocket will download where the cavity is. It will give the cavity formation. Let's just download them, save them into the Bioinfo workshop folder. So yep, let's save like this queue pocket. Something like this. Okay, so click on save. If you open the file as is, it can be very confusing. So we should open it using Excel. I'll show you how. Open a blank Excel workbook, click and drag the text file to here, and then we got something like this. And then let's make it bigger. Okay, we can click on data, then we tap some column, and then keep on delimited, and then enable space. You can see how now it differentiates all the columns separate very nicely. So we can report our binding cavity, this one, as the potential ligand binding site of our protein of interest. The most important part is the cavity size, the cavity volume, and then also the ligand binding score. The more negative it is, the stronger the ligand binding affinity. And also this here is the contact residue. Here's a fun fact. This contact residue is telling us, for example, um, LYS 72 and A. It refers to lysine residue 72 on chain A. But if you want to make it much easier for readers to understand, you can copy it and paste on ChatGPT. Let's go to my chat GPT. We copy it like this, and then we write something like this: convert LYS 72A to LYS 72, and so on. So now it is much easier for people to understand. So we can copy it and then go to Excel, paste it. So now it looks much easier. Then you can save the file as Excel. So you can save it as Excel. So now we have our complex files and also our 5UJV chain prep. So we're going to open it using BioVR. Let's open the best one first with the highest ligand binding affinity. We're going to like this. We press Ctrl H to open the hierarchy, or you can click on View and click on Hierarchy here. Okay, so this is a predicted. This is our PYL10, and this is one of the predicted ligand binding position. In practice, we're going to do something like this. We're going to bring in all the others by simply click on it, make it smaller. We drag and drop like so. Yes. 1, 2, 3. I think we're missing this one. Yes. Okay. So what we're going to do is we're going to label each one according to the label here. Okay, so this one should be this one. So I'm going to write C2 7.2. It's kind of weird we have two C2, but nevertheless, this one should have been C1 really. And then write them C2 here, and we're just going to write C2 for the sake of simplicity. And then we're going to do the same here, C3, same here, C4, same here, C5. And then here's a bit tricky part. We're going to click on C2, click on C2, control, click on C3, control, click on C4, control, click on C5. And then we're going to drag them to here. Oh, sure. Okay, so we have to do one by one, unfortunately. Let's do it one by one. C4, C5. Right. So now all the dot position is in one complex. So you can click this here, hold shift, and click here as well, and you can delete it. And yeah. Oh, if you did something wrong, you can click on Ctrl Z to undo. Ctrl Z again. Ctrl Z again. Again. Ctrl Z again. See, you get our files again. So Ctrl Z to undo. So you can delete it by pressing delete. So now we're going to save this as, oh sorry, we're not going to save it just yet. So now we're going to bring in 5UJV chain A prepped to here. So this would be our, oh, let's rename our protein as well, so it's easier. L10 and 5UJV, and here ref AB, reference AB. All right, So as you can see, we can color them differently, so we don't get confused. Let's change this color to white and change this color to, let's go with light pink. Okay, lovely. So you can see it's very almost identical because the RMS is very low, so that's good. So now we're going to find the reference ABA, and then let's change the color as well. Let's give it um, light green, lime green, yeah. Okay, if we zoom in by using the mouse wheel, you can see that there's a very strong overlap with this one. So this is C1. So as we predicted, the C1 shares a very strong overlapping with the reference. So C1 is likely our ligand binding position on PYL10. So that's our so this is our binding is our active site, which we will report in the Q pocket data that we have. So again, let's go for the Q pocket data. So we're going to report this as our ligand binding site. Let's give you a book. All right. So yep, that is done for molecular docking. All we need to do now is simply mutagenesis. Okay, now we're going to finally perform mutagenesis. We're going to load the PYL10 to camera for practice. Let's use the normal camera. We're going to click favorites, and we highlight a particular sequence that we want to manipulate. Then we're going to click tools, structure editing, rotamers. We're going to select a sequence, and then we convert it into a desired rotamer. And then we select the whatever substitution that we perform. We're going to select the rotamer with the highest probability, and then we need to save the mutated protein as .pdb. Right? Let's do it. Okay. Just open the earlier camera file. Okay, let's just work with the one we have earlier. But if you have closed the UCF camera, that's fine. Just open the PDB again. Double click and open. Okay, let's go to model panel. So it doesn't look messy. I'm going to hide the 5UJV. So now we only have PYL10. We click on favorites. We click on sequence. Then we sequence for PYL10. Click on show. Okay, now we have the sequence. Right. Say for example, okay, I want to manipulate this AA. We can do one by one. We can do multiple as well. So we can convert everything to, for example, alanine, or everything, all of this to alanine at the same time, or we can just do one. For this exercise, let's just do one at a time. So this is going to be here. Okay, let's check it out. So that part here is alanine. Let's click on actions, atoms, and show. Okay, we will show the residue here. So alanine is a very small amino acid, so we only have very small functional group. So that's why it looks like this. So let's mutate it into something bigger, like phenylalanine, which is big, benzene ring. So we go for structure editing, go for rotamer, and then from here, the atom type change to phenylalanine here, and then we click on OK, and then this is what kind of configuration you want to have for the phenylalanine. For the sake of simplicity, we're just going to pick the one with the highest probability, which is the first one. Then we're going to click OK. So now it has changed to phenylalanine. Right. So we can just find it, click on File, Save PDB, and then click on the save model PYL10, and then we just write PYL10 return. Okay. Save. Okay. If we point our mouse here, it's written is phenylalanine 20. Before phenylalanine, it was alanine. So let's see if the mutation is correct. Let's go back to our workshop folder. Open the PYL10 mutant using BioVia. Okay. We press Control H. Okay, let's check out 20. See, it's phenylalanine. If we look into the PYL10, the original ones, if you look at 20, it's alanine. So we have done it correctly. So that's it. That's all for part 1 of this tutorial.